and good evening to everyone. I do have a single disclosure. I am the founder and CEO of Sequence to Script Incorporated. This is just a company that provides laboratories with translational services, pharmacogenetic testing results into clinical recommendations. There are three learning objectives for the presentation today. So hopefully by the end of this session, you'll be able to explain the rationale and evidence for using pharmacogenetic guided prescribing, identify care situations where pharmacogenetic testing could be useful, and then finally describe some of the considerations for using pharmacogenetic testing in your practice. So I like to start off all my talks with this quote. I really think it captures quite nicely the current state of prescribing right now, but also highlights the need for more precision or personalized prescribing. If you look at this in another way, at the population level, we know that medications are efficacious and tolerable. But if I take someone at random out of that population, it looks a bit more like this, where efficacy and tolerability vary quite a bit. And so we're left with this challenge of determining which drug fits in which one of these quadrants for a particular individual. And so this really highlights that need for personalized prescribing strategies. So here I've just listed a few of those. Some of these are quite familiar to everyone on the call. They've been around for decades and they're considered standard practice. Today, of course, we're going to be focusing on the new kid on the block for these strategies and pharmacogenetics. But before I get started, I just want to make it clear up front that pharmacogenetics isn't going to replace any of these strategies. It's meant to complement or enhance these strategies and should be used in combination when applicable. So what is pharmacogenetics? I know you heard, if you attended the first session of this series, a bit about this already, but just to review this again, pharmacogenetics uses genetic information to predict a person's ability to process and react to medications. And we typically divide this genetic information up into two broad categories, pharmacodynamics and pharmacokinetics, which I'm sure everybody on this call is very well versed in. I'm just going to say here that to date, the evidence is mainly been focused on pharmacokinetics within the field of pharmacogenetics, although there is active research going in pharmacodynamics as well. But if we're looking at what's ready for prime time, what's ready for the clinic, it's primarily genetic information that's coming in that pharmacokinetic space, and more specifically metabolism. So when we think about metabolism, we often will think about drug metabolizing enzymes. And thankfully, we have some really good pharmacokinetic studies that have been done that have looked at the association between genotype data within these drug metabolized enzymes and correlated them with pharmacokinetic outcomes so that we could get an idea of the enzyme activity without actually taking blood. We can just do the genotyping through saliva, or like a buccal swab or saliva sample. And so what we know is that we can predict phenotype, and this typically is one of these 5 phenotypes when we're thinking about drug metabolizing enzymes, ranging from poor to ultra rapid. And if we know someone's predicted phenotype, we can then reasonably predict how much of that drug will be in their blood if given a standard dose, with those on the poor intermediate side of the spectrum being at higher risk for side effects, and those on the rapid, ultra rapid side of the spectrum being at higher risk for no benefit to the drug. Now, of course, we have some of this good pharmacokinetic data, but which drugs, which genes do we have this information for? And so we really do need to just quickly review the evidence and where that evidence comes from. And like any other field of study, we want to look at the published literature, but in pharmacogenetics, we also are very much interested in the product labels or the drug labels. Now, fortunately, we have a group known as the Pharmacogenomics Knowledge Base, or PharmGKB. They're based in Stanford and they're NIH funded. And what they do for us is they pull out all these gene-drug associations from the literature and the drug labels. They curate that information, and then they score it on the strength of evidence scale. And the way they do this is an unsupported gene-drug pair is one that, as the name would suggest, the evidence doesn't support that association. A low strength of evidence are those gene-drug pairs that have maybe a single study that has shown an association, or there's a number of studies, but there's mixed or conflicting findings. When we get to the moderate level of strength of evidence, that's where we're starting to see the multiple replications. But we could argue that these associations, even though they've been replicated, are of small effect and may or may not have any clinical relevance. And then finally, the high level of evidence, which is what we're obviously most interested in, is where we see multiple replications and effect size that's large enough to be clinically impactful. And it's these gene-drug pairs that get pushed forward into the guideline development pipeline. And I'm showing here 4 of the major guideline developing groups or expert groups. The top 2, CPIC, or the Clinical Pharmacogenetics Implementation Consortium, and the Dutch Pharmacogenomics Working Group, are probably the 2 largest and most well-known guideline developers, and they've created the most guidelines between them. But there is a French group, and there's even a group here in Canada that creates guidelines, just so that you're aware of. Now, all together, collectively, these guideline developers have created prescribing guidelines for 115 gene-drug pairs that comprise 25 genes and 88 drugs. And those are shown here in this, I know, rather busy figure. The reason I like putting this up, though, is because I think it really shows in a nice snapshot that it's no specific area of medicine that pharmacogenetics is impacting, that it really impacts a number of areas. It's not just one. And so this is, I think, a nice visual to keep in mind when thinking about pharmacogenetics. Now, just to give you a bit of a case example of why this is potentially important and how it can be useful clinically, this is a 67-year-old male that had a diagnosis of major depressive disorder. And he was admitted to hospital with suicidal ideation and catatonic symptoms. And then prior to his admission, he was given trials of citalopram and escitalopram at maximum doses, but showed minimum response. So the treatment team decided, well, let's start ECT, electroconvulsive therapy. And his mental state did improve after ECT, and then he was prescribed sertraline prior to discharge from the hospital. However, shortly after his discharge, his depressive symptoms reoccurred, and he was readmitted. And this is when the team decided to order pharmacogenetic testing. So here I'm showing you the results of that testing. You can see for this gentleman, he was a CYP2C19 rapid metabolizer and CYP2D6 normal metabolizer. So let's just unpack this a bit and see what this means for this gentleman's treatment. So we're looking at the evidence and guidelines. First, let's just state that all 3 drugs that he was trialed on were CYP2C19 substrates. And so that's just important to remember. When we look at the drug labels, we see that for 2 of the 3 drugs that he was prescribed, citalopram and escitalopram, the drug labels do mention CYP2C19 genetic variation and how that may affect the pharmacokinetics of these 2 drugs. And if we look at dosing guidelines, there are dosing guidelines developed by CPIC for all 3 drugs, and they specifically state that an individual that is a CYP2C19 rapid metabolizer, they should consider an alternative drug not predominantly metabolized by CYP2C19. And then finally, if we look at the PharmGKB strength of evidence scale, all 3 of these drugs in relation to CYP2C19 are rated as high, again, because of those multiple replications and a large effect. So in the end, this gentleman sertraline was discontinued for him, and they started paroxetine. And if you recall, he was a CYP2D6 normal metabolizer. Paroxetine is primarily a CYP2D6 substrate. And so this would be a good choice from a pharmacogenetic perspective. And he was discharged 3 weeks later, and he remains well in the community with no further relapses. So the take-home message for this case is that pharmacogenetic testing could significantly reduce these types of experiences. Also could expedite the time to response. As you recall, he had done 3 trials of an SSRI before finding on the 4th trial the one that actually worked for him. And it may also avoid unnecessary treatments. In this case, he was exposed to a round of ECT, which could arguably be said that he didn't need that if paroxetine was started sooner. So there is a good amount of consensus to suggest that pharmacogenetic testing is clinically useful, and that the evidence base is there, but adoption of pharmacogenetic testing is still modest. And I would say that for the most, it kind of depends on which area we're talking about, but for the most part, we're in this innovator or early adopter phase if we're talking about this diffusion of innovation curve. And so anytime we're talking about a new innovation or a new treatment or anything, all of these things need to cross what they call the chasm. If these innovations can't cross this chasm into the early majority, they basically just fizzle out, and people move on to another innovation or go back to the status quo. So the big question we have in our field is, can we cross this chasm? And in order for us to do that, we really will need to address some of these implementation considerations that I'm going to speak to you about in the next part of this talk. And I've summarized them here in these circles, and I'm going to touch on each one of these briefly. So the first consideration is related to selecting and ordering. And by far, the number one question I get asked is, what test should I use? And it's a simple question, but it's not that simple to answer. It often results in me saying it depends. It does depend on the indication and where you're testing and what you're really looking for from the testing. But because we get asked this question so often, we did develop this guide to test selection, and we created this decision tree. Now, I know the decision tree is too small for you to read, but basically what I'm putting it up here for is so that you know that it exists and that you can go to it if you want, or feel free to email me and I'll send you a copy. But I'm going to boil it down into the 4 basic components of that decision tree, or what we call the best practices. The first is the test should be evidence-based. That seems like a no-brainer, but I'm going to say it anyways. Going back to that complex figure that I had with all of the gene-drug pairs that have guidelines, we want to select tests that are based in that evidence, that have prescribing guidelines. Secondly, we want to select a test that's being performed by a laboratory that's accredited. This ensures a high level of analytical validity. Third, we want the testing laboratory to be transparent about what they're testing, how they're testing it, and the process they're using to translate the genetic results, so the genotypes, into prescriber recommendations. If they can't tell you those processes or how they're testing or what they're testing, I would suggest moving on to another provider. And then finally, the testing needs to be accompanied by some type of support, clinical decision support. This could be in the form of a portal that helps with decision-making, a paper report that has more descriptions about the test results, or ideally, some kind of linkage to an expert in the field of pharmacogenomics that can help with some of the more tricky questions around interpreting and implementing pharmacogenetic testing in practice. And the last thing I want to say about this particular topic is that no test that I've seen is superior to all other tests that I've seen. But there are inferior tests, and these are the ones that we want to be cautious about and, if possible, avoid. The second component of the selecting and ordering, and probably one of the other commonly asked questions that I get, is when is the best time to order a test? And again, this also is not as simple to answer, but it's a bit clearer than what to test or what test to use. There are three main approaches when thinking about when to order. The first is a reactive approach. So this is when we're ordering pharmacogenetic testing after an individual has had a poor response or series of poor responses to a medication, or has developed side effects that are intolerable. And this is probably the most commonly used approach to pharmacogenetic testing. And in fact, in the US, this is the approach that is being reimbursed at the moment. The second approach is the point of care approach. So this is when you order pharmacogenetic testing at the same time that you're making that initial drug selection and dosing decision. So the testing won't necessarily help with that initial decision-making around which drug you're going to select and how to dose. But the notion here is that the test results will be available soon into or during the titration period, I guess depending on which drug you're using, but adjustments can be made earlier. So you're not necessarily waiting to see if they respond or not or have side effects. You may be able to make adjustments before those occur. And then the final approach is the preemptive approach. And those of us in the field, most of us in the field, are proponents of this. And as the approach name suggests, you're doing or ordering pharmacogenetic testing prior to that initial drug selection and dosing. And so that if it's available, you can use that information along with other information that you normally would to help make that initial drug selection and dosing. I will say that currently I'm not aware of any program or place that's doing this on a regular basis, but there are places that are starting to implement preemptive testing more and more as time goes on here. Okay, now switching gears a bit to another consideration, which is the content validity of these panels that are offered by different laboratories. So you may or may not know that regulatory standards for what a laboratory puts on their panel have not been established. So right now, there's nothing keeping a laboratory from putting any gene on their panel that they choose to. And so what this leads to often is cautionary content. And so what I mean by cautionary content are genes that have good face validity, good biological plausibility, but really when we're talking about clinical validity, it's inconclusive. So I have a few examples. These examples are based more in psychiatry. That's just the area that I work in the most. But they apply, there's cautionary content and cautionary gene-drug pairs across different disciplines. So if we just take, for example, the first one here, the SL6A4, which is the serotonin transporter gene, and SSRIs. So obviously, great face validity, good biological plausibility. But when we look at the evidence, there's still a lot of inconclusiveness around the clinical validity of using the serotonin transporter genetic information to prescribe SSRIs. And I can say the same for all of the other three that I have here. I'm not going to go into each one, but all three have good face validity, good biological plausibility. But when we look at the clinical validity, it's just not there yet. Not to say that it won't be in the future, but right now, it's not there. And so to give you an idea of how often this cautionary content occurs, we examined, I think it was 25 panels that are focused on psychiatry. And you can see here that it's a non-trivial amount of these panels included these cautionary gene-drug pairs. Now, I'm not saying that these panels shouldn't be used necessarily, but when interpreting the results of these panels, these particular gene-drug pairs, I would be cautious about taking action clinically. and maybe ignoring this and focusing on the content that actually has guidelines associated with it. And the reason for that, I can illustrate with this second case. This was a 25-year-old male diagnosed with schizophrenia. He was admitted to hospital on an involuntary basis due to bizarre behavior and command hallucinations. He had a history of multiple medication failures, and because of that, the treatment team decided to start clozapine. Unfortunately, the patient's family and family doctor opposed the decision to start clozapine based on pharmacogenetic testing that the patient's family doctor had ordered previously. So here is the summary of the results that the family doctor had ordered and got back from the testing laboratory. And you can see for the most part, the test results are unremarkable, but I've starred the key finding here. So this particular laboratory tested for the D2 dopamine receptor gene and a particular variant on that gene. And what they said is basically for this individual, his genotype suggests that he'll be a poor responder, and they specifically highlight clozapine. So based on this finding, the family and the family doctor were opposed to starting clozapine. So let's unpack this a little bit and look at the evidence for DRD2 and clozapine. So first, if we look at the drug labels, product labels, we see that there's no mention of DRD2 genetic variation on any of the clozapine labels. There's also, to my knowledge, no dosing guidelines associated with DRD2 or clozapine at the current time. And if we look at the PharmGKB strength of evidence scale, this particular gene-drug pair is ranked as low strength of evidence, which means obviously not yet replicated or conflicting findings. In this case, there's a lot of studies that have looked at DRD2 and clozapine, and so this is more around conflicting or mixed findings of this. So there's really no consensus on it. So in the end, for this particular case, they did commence clozapine after getting approval from the medical director, and the patient consented. And the patient responded rapidly and was discharged. And he gradually returned to his previous level of functioning. So the take-home message here is that we really need to be careful about straying away from gene-drug pairs that do not have prescribing guidelines. When we stray, we put ourselves in situations like this where we may be taking an extremely efficacious medication off the table for a particular patient. On the flip side, we may also end up inadvertently prescribing something that was not a very good match for the individual based on the pharmacogenetic results. So it's just important, and I can't stress enough, that we stick with those prescribing guidelines from those expert groups. And then one final consideration I want to say about content validity. So I was talking about the gene content, but even if a panel has all the genes that have guidelines and nothing else, we still need to take into consideration the alleles within that gene. So what are they actually measuring? Because certain populations or certain individuals may not be well represented by the alleles that they're measuring in that gene. And just to give you a bit of an example here, I've just selected CYP2D6 as an example in 3 of the many alleles in that gene, just to show you how it can vary from population to population. So you can see here the star 4 allele more or less is similar across populations, a little bit different in some, but for the most part, very similar. But then when you look at the star 10 and star 29 alleles, you see this vast difference based on ancestry. So individuals of an Asian background are much more likely to carry the star 10 allele, and individuals of African descent are much more likely to carry the star 29. So if you have an Asian patient and you're looking at a panel and the panel doesn't include the star 10, that would be a potential problem because that could result in erroneous findings. And likewise, if you had an African patient that didn't have the star 29 on the panel that you were thinking of, that also could be a problem. Now, thankfully, the Association for Molecular Pathology has created these guidelines for laboratories. They call them the must-test alleles. And I've just put up, they've done more than just these 3 genes, but just as an example, because these are the basically 3 primary genes that are used in pharmacogenetic testing. They've created this must-test list, or tier 1, to help laboratories avoid this problem. But it's always good to check, or at least ask, are you measuring the tier 1 alleles for these genes? And the reason I say that, I'm going to just give you a quick example here. Let's say we have an individual that we know is a CYP2D6 star 10, star 29. So that we know that that's their genotype. But just as an example, we have 2 laboratories. Both laboratories are testing for 12 CYP2D6 alleles, but they have a different composition to their panel. So lab 1 is testing star 10, but they don't have star 29, whereas lab 2 has both of those alleles. So when they genotype this individual, lab 2 is going to get the correct genotype, star 10, star 29. Whereas lab 1, they're going to be able to call that star 10, but because they don't have star 29 on there, and particularly this variant here, they're going to default to a star 2, because star 2 is defined by these 2, which they do have. So what does that mean? Well, lab 1 is going to call this individual a normal metabolizer, but lab 2 is going to correctly call them an intermediate metabolizer. So this potentially has implications for prescribing recommendations depending on the drug, but this is why this is an important issue, and that we need to make sure that these must-test alleles are included on these panels. Okay, now switching to clinical efficacy. I'm not going to spend a lot of time on this. As I mentioned, the evidence is actually quite strong from many areas. There are meta-analyses of clinical trials in a number of fields that support the efficacy of pharmacogenetic testing, from antidepressants to anticoagulants, analgesics, proton pump inhibitors, and the list goes on. And I have only listed a few here. But there is a strong evidence for its use. What has been more recently kind of more challenging is determining or demonstrating the cost effectiveness of the testing. So we know it's efficacious, but is it cost effective? Well, most of the evaluations do suggest that pharmacogenetic testing is cost effective or cost saving strategy when compared to standard of care or non-pharmacogenetic testing guided prescribing. And that's shown here in this pie chart. You can see if you add up the 2 green ones, which cost effective or cost savings, you see the majority of studies do show that it is cost effective or cost saving. And if we give a specific example in mental health, like I said, the area that I work with most, we see that individuals given or provided pharmacogenetic guided prescribing, we see fewer hospitalizations, emergency room visits, and visits to healthcare providers, which of course translates to overall cost savings. In this case, 3 independent studies have shown somewhere between $6,000 and $7,000 US per patient per year is saved by using pharmacogenetic guided prescribing. Okay, now on to feasibility. Now, this probably from a clinical perspective, this are some of the biggest challenges when we want to implement pharmacogenetic testing in a practice. One of the big ones that's talked about a lot these days is the integration. So how do we integrate pharmacogenetic testing into current EHR and EMR systems? Few institutions globally have actually accomplished this. There are a few, but very few. And in Canada, I'm not aware of any that have done it completely yet, although there is movement towards that. So of course, this is a challenge. The healthcare provider that orders the testing typically knows that it exists, but nobody else does. And so there is this problem with integration and sharing of the results across different providers. Another big challenge, and this learning series is addressing this challenge, but we need more of this, is that healthcare providers' confidence in their pharmacogenetic knowledge is typically quite low. And it's probably the most cited and consistent hurdle identified across studies that have looked at barriers or hurdles to implementing pharmacogenetic testing. So we all know if we're not very confident in doing something, we're probably not going to do it. And so one way, obviously, to boost confidence is to have more education around pharmacogenetic testing and obviously incorporating it more in training programs, which is starting to happen. And then finally, probably, which is always the elephant in the room, is the affordability. So it's a wide range of costs for these tests. If we focus just on Canada, the variability is a little less. The median cost of pharmacogenetic testing in Canada is about $500. And there's variable reimbursement. That's globally, but also here in Canada, it's very variable. There is some scenarios or some schemes that can get you reimbursed, but not everybody has access to those. And the other thing I want to mention about affordability is that the test cost does not correlate with quality. So we've looked at this, and it's clear that the more you pay for a test does not necessarily mean it's a higher quality test. So in this case, in pharmacogenetics, you don't always get what you pay for. So definitely don't base the selection of a test on the price of the test. You really need to look at some of those other factors that I've already mentioned. Okay, and then the final consideration that I wanted to share with you today, and probably, in my opinion, the most important from a clinical perspective is how do we interpret this test and implement it. And of course, we know that pharmacogenetic test results can be quite challenging to interpret, particularly when we don't have accompanying clinical decision support. As I mentioned earlier on, the best practices when selecting a test, we really need to make sure that there's some type of clinical decision support. If we just hand healthcare providers genotypes, or even a phenotype for that matter, like you're a normal metabolizer, it's unlikely that many will know what to do with that. So we need to have complete support, wraparound support for pharmacogenetic testing. So what we did a while back actually was we wanted to look at the different decision support that's happening with these commercial companies. And so we did a head-to-head comparison. It was a small study. We looked at 5 individuals. We took saliva samples from each of these 5, and we sent saliva samples to 4 independent laboratories. And then when we got the results back, we compared within and between these individuals to see how much concordance there was in the results. And what we found was there was less than 60% agreement in the advice or the recommendations that were being given across tests, even though it was the same exact individual. So this led us to believe that, we can't assume that these clinical decision support tools are equivalent or interchangeable, and there is definitely a high bit of variability. So why is this? Well, many of the tests that are out there, not all, but many, use proprietary algorithms, or what we call black box algorithms, or sometimes it's referred to as what's the word I'm looking for? Anyway, they use decision algorithms that you can't really reverse engineer. So it's a bit of a black box, like I said. The problem with that is they often will tell us what's in the box, but we don't know exactly what proportion of each ingredient they're using. And so we know that they may be using the CPIC guidelines, and they mentioned PharmGKB, and they may even reference some of the published literature, but we don't know how they're using that specifically to make these medication dosing and selection recommendations. And so this becomes an issue, and this is why we get this discordance when we look across the different tests. So one solution to this is that we can bypass that black box by just taking the raw genotype data that all reports will say. They'll give the genotype, and just applying that directly to the guidelines, which are all freely available, right? You can go to PharmGKB and you can type in the patient's genotype, and it'll provide you the recommendation for a particular drug. Now, not many healthcare providers have that kind of time to go through and look at all the different drugs that they need to look at for a particular individual, for maybe numbers of genotypes for different genes. So this becomes a tedious task, and most people won't do it. So what I mentioned in my disclosure is we developed this sequence, this script. It's a free tool for physicians and pharmacists and healthcare providers to use to help bypass that black box. So basically, you can enter the genotype information of the patient from any report that you receive the information from, and it basically does a little bit of a second opinion, you could say. It only uses guidelines. So there's no black box. It takes directly from the guidelines. It's just making it easier for you. You don't have to go back and look at the FDA, the Dutch Pharmacogenetics Work Group, and CPIC or PharmGKB. It's just putting it all in one spot, and it creates a report for you. Now, sometimes the report will say exactly what the commercial laboratory said, but sometimes there'll be some discordance. And so that's kind of the flag that they may not be following strictly the guidelines by these expert groups. And this is just one tool. Of course, it's the tool I'm most familiar with. There are other ones out there. But again, this is a free tool. I'm not trying to sell you anything. And the last thing I want to talk about when I'm talking about an interpretation has nothing to do with genetics, really. It's more about the non-genetic factors and how they can impact how we interpret the pharmacogenetic test results. And this is often overlooked when we're talking about pharmacogenetic testing. And this is a phenomenon known as phenoconversion. So this is when there's a mismatch between the genotype predicted phenotype, so the metabolizer status, and what's clinically observed as the phenotype. So let me give you an example. So if we think about phenoconversion, we're typically thinking about the presence of an inducer or an inhibitor of a particular enzyme. So let's take CYP2D6 as an example. Let's say we genotype someone, and based on their genotype, it comes back that they're a normal metabolizer. But then we look at their drug list and we see, oh, they're actually they've been taking for the last 6 months, they've been taking fluoxetine. We know fluoxetine is a potent CYP2D6 inhibitor. So even though their genotype suggests they're a normal metabolizer, because they've been taking fluoxetine, they're likely going to be, almost guaranteed they're going to be a poor metabolizer, assuming there's not an inducer or something on board. So this is something we need to keep into consideration. And just to give you an idea of how often this occurs, there was this nice study done on 900 patients treated for depression. And the first thing they did was just genotype them and determine what proportion were CYP2D6 poor metabolizers. And so they found just genotype alone, 4% were poor metabolizers. And this is exactly what we'd expect. This was a European-based population. And in European individuals, we'd expect somewhere between 4 and 6% of people to be poor metabolizers. So this is right where we expect it. But then they looked at their current drug list, so what drugs they're currently taking, and estimated what proportion of them would be poor metabolizer phenocopies. So again, phenoconverted to poor metabolizers. And there you see there was 27% that actually would show clinically to be poor metabolizers, even though genetically only 4% were predicted to be. So this is obviously huge discrepancy. This is something that we need to take into account. And if we don't take this into account, it can lead to erroneous action. Okay, so I'm going to finish off. I have 3 take-home points. The first, which I hope I've been able to convey to you, is that pharmacogenetic testing is an evidence-based strategy to inform prescribing of several commonly used medications. 2, when we're using pharmacogenetic testing, it's imperative that we adhere to the available guidelines, and certainly don't forget about the non-genetic factors and phenoconversion, like I just described in the previous slide. And then finally, that pharmacogenetic testing, as I mentioned in one of my first slides, is a companion decision support tool. And it's really meant to enhance, not replace, current prescribing strategies that you're using in your practice. Okay, I'll stop there, and I look forward to any discussion or questions that you may have. Thank you.
Great. Thank you so much for that presentation. Such a clear presentation on what is clearly a very, very complicated subject. So thank you for that. If you do have questions, please try to use the Q&A feature. We do have one here. So someone asked, how do we know what is a cautionary pairing if we're not specialists in this field?
Yeah, so basically anything that doesn't have a CPIC or Dutch Pharmacogenetics work group kind of guideline, basically. So you remember that busy figure I had of 115 gene drug pairs. If it's not on that figure, I would consider that probably a cautionary pairing. I will say that that figure did not have the FDA drug label worked into it, so there is some caveats there. But from a prescribing guidelines perspective, if it's not one of those, that's what I would define as cautionary.
Okay. Someone else just said thanks for the presentation. Thanks for joining us. Yeah, so if you do, oh, here another one came in. Will the genetic testing only need to be performed one time for one patient and consider the cost to be a one-time expense?
Yes, but there's caveats. It depends on it depends on what was tested. So let's say you the first time you test the patient, the panel that you test for only has, let's say, CYP2D6 and CYP2C19. That's great. That covers a lot of the drugs, but the evidence keeps going, and there might be new genes. Or let's say, oh, I want to prescribe an NSAID, and that guideline is CYP2C9. I didn't order CYP2C9, so now I have to test for that. So there is a potential that it's not a lifetime test. For the genes that you tested for, yes, but you may have to retest for genes that evidence comes out and they're not on the panels yet, right? So it's a yes and no kind of answer. Sorry for not being, I can't be for sure about that, but for the most part, it is a lifetime test if it's for specific gene.
Can we conclude to be reactive? Sorry, can we conclude to be reactive in case of lack of prescribing guidelines? So is it reactive if there's no prescribing guidelines?
I'm not sure I completely understand the question, but is it reactive if there is no prescribing guidelines?
If that was your question, if you just want to clarify at all.
Yeah, I don't want to read between the lines here, but yeah, I mean, if there's no prescribing guidelines for a particular drug, I'd say, and that's the reason why you're testing, I'd say don't test. There's no need for that in a reactive standpoint. Of course, you can always test if you're thinking preemptively, like they're going to need more drugs in the future, let's do it now. Completely support that. But if you're like, we're having trouble, this is the drug we're thinking of, we want to go with this, we want to see if pharmacogenetics can help, there's no guideline for that drug, I wouldn't bother in that particular scenario.
What courses are available in Canada for pharmacists who are interested in pursuing this as a career? To sort of add to that, are there any other resources you might recommend, maybe not just courses? Yeah,
I mean, other than the pharmacy schools themselves, which are now incorporating pharmacogenetics into their curriculum, I don't know if there's standalone courses. I should say there definitely probably are, but I'm not aware of specific for pharmacists. Yeah, they're still kind of coming online here. Like most of the education that's occurring in pharmacogenetics is done through learning series similar to this, and different pharmacy organizations kind of sponsoring those kinds of things. So yeah, it's a big need. Like I said, about feasibility, this is where we need to move. Yes, I just see University of Florida does have a course. There's a couple others as well. I don't know accessibility with University of Florida. If it's like you have to pay their tuition, or if it's I'm not sure how that works, but I think Vanderbilt also has one. So yeah.
And another comment just about there are some textbooks as well, they said.
Lots of textbooks. Yeah, not all are great though, so be careful with those. If you really want to buy a textbook, email me and I'll provide you some recommendations, because I've ordered most of them and some of them I've been thoroughly disappointed with. Some of them I've been very happy with. So.
Does testing have to be performed on a referral basis? Physicians are the only ones to access or request the test from the lab, or is the service public access from the labs?
Yeah, so it depends on the lab. Some labs require physician involvement. Some just require a healthcare provider that would include a pharmacist, and some will allow direct to consumer. So it really just depends. It's really, I would call it the Wild West right now. Because there's really no regulation there either. So like things like even 23andMe is now including pharmacogenomics as one of the reports. I don't think it's available to Canadians, but in the US you can. So you just have to look at the particular laboratory that you're looking at and see what's their ordering process, because they all are a bit different.
Someone asked about your opinion on PGX Academy. Are you aware of it, and what do you think?
I have heard it. I haven't gone through it, or I don't have a opinion right now, I should say. I don't know enough about it to say, but I have heard of it. I don't know if that means anything. It's kind of come around, yeah. So.
Okay, so no questions right now. We'll maybe just give them like another minute or so. If you do have a question, please put it in the Q&A. If you think of a question after, feel free to email us at education@opatoday.com. We can reach out to Dr. Bausman and pass on questions to him. Again, if you want to join us for the third part in the series, it is coming up on August 25th. We'll just give them one more minute if they have any questions. Do you have any final words? I know you had like your takeaways, or they were summarized, but like any advice that you would generally give if someone's looking to like get into the service for their patients?
Yeah, I mean, I think the biggest advice I always have is, make sure that you're testing for the right reasons, I guess. Like I said, I'm always preemptive, but right now it's at a cost to the patient, right? I mean, we're trying to move more towards equitable availability of pharmacogenetic testing, but until it's reimbursed, that's not going to happen. And to my knowledge, no province has included it in their healthcare delivery. So I think you have to keep that in mind when doing it. So if you're going to test, make sure that there's a good reason for it. And like I said, I just follow the guidelines. I mean, they're there for a reason. Yeah, they're conservative. So, it takes a while to make a guideline. So sometimes people say, oh, the evidence is there, but there's no guideline yet. It takes time for these guidelines to be made. So I'm not saying that there's not things in the process and the pipeline that don't have a guideline now, but we'll have later. But for the most part, I think it's okay to play conservative, particularly when people are paying quite a bit of money out of pocket for these tests.
Okay, so we have another question. Is it therefore safe to assume that aging has no impact on genotype expression?
And it's not safe to assume that. So certainly there's still a lot of work to be done on that, but what I've seen so far, it depends on the gene or the enzyme. So some enzymes don't seem to be affected much by age, like CYP2D6, let's say. It comes online pretty quick after birth, and it stays at adult levels throughout life. Whereas other ones like CYP2C19, it has higher expression than adults early on, like in early childhood and early adolescence. Then it comes back down to adult levels, and it just keeps going. after 65, it really starts to peter out. And so genotype testing and those and the extreme ends of the age spectrum, we typically don't do too much pharmacogenetic testing at the early ages. That could change, but definitely in geriatric populations, when the medications, obviously lots of medications, this is something you definitely would want to consider. A normal metabolizer that's 78 years old may not be a normal metabolizer, just because the genetics says they are. So I wouldn't assume that. In a 40-year-old adult with no hepatic or renal problems, yes, you can assume that. But you know, everything else, no one's completely normal, right? So you have to keep all that in account. That's why I said it's a companion tool. So we have to keep that into consideration.
So yeah, so no more questions. So I will let everyone go for the evening. Thank you all for joining us and have a great night.
Thank you. And feel free to reach out.
Bye.